So good morning, sir. I am the presenter of Group 1, and our topic is about digestive system. First article is about the morphological study of the alimentary canal and malpighian tubules in the adult stage of pollen beetle meleagetes. So for the introduction, the study examines alimentary canal and malpighian tubules of meleagetes chinensis and agricultural pests. So these organs are essential for digestion and waste removal. So understanding their structure helps explain the insects' feeding behavior and supports pest management strategies. So for the summary, the researchers used dissection and microscopy to study the insects' internal organs. So, ang alimentary canal is divided into foregut, midgut, and hindgut, and a well-developed midgut for digestion. So, the malpighian tubules are numerous, indicating efficient waste excretion. For the critical analysis, the study provides clear and detailed anatomical descriptions, making it useful for research. But, however, it is limited to morphology, lacks functional analysis, and focuses only on the adult stage without comparison to other species. So for the discussion, the structures are well adapted for pollen feeding, allowing efficient digestion and waste removal. So these adaptations help the insect survive. Further studies should include physiological and ecological aspect. So for the conclusion, the study offers important insights into insect morphology and adaptation. However, more comprehensive research is needed to fully apply the findings to pest control. So for the Article 2, morphological structure of salivary glands, alimentary canal, and malpighian tubules in adult Erythema spectabilis. So this article focuses on the anatomical features of an agriculturally important insect. So for the introduction, the study examines the internal structures of Erythema spectabilis using microscopy. So it aims to describe the digestive excretory systems and explain how these structures help the insect survive and feed on plants. So as a summary, the article explains that the salivary glands produce enzyme for digestion, while the alimentary canal is divided into parts responsible for food intake, digestion, and waste formation. The malpighian tubules function in excretion and regulation, so these systems work together to support the insect's feeding behavior. So critical analysis, the study is strong in providing detailed and well-organized anatomical descriptions that are useful in pest biology. However, it is limited because it only describes structures without experimental data and lacks quantitative measurements and provides minimal comparison with other species. So the discussion, the findings are important in understanding how insect body systems function and adapt. So this knowledge can help in developing pest control strategies and serves as a foundation for further research in insect physiology. Conclusion: overall, the study is clear and informative with strong anatomical detail. Although it lacks functional experiments, it does still contribute valuable knowledge to insect morphology and can support future scientific studies. So for the Article 3, the antibiotic treatment impairs protein digestion in honeybee, Apis mellifera. For the introduction, the study investigates how the antibiotic oxytetracycline affects protein digestion in honeybees. Protein is essential for bee growth and colony health, making this research important in understanding how antibiotic use may impact bee nutrition and survival. So for the summary, the researchers conducted an experiment using newly emerged worker bees placed in cages and fed artificial diets with varying protein levels. Some groups were treated with antibiotics while others were not. So results showed that antibiotics did not affect survival or food intake but significantly reduced protein digestion efficiency, especially in bees with low protein diets. So this effect is linked to disruptions in gut microbiota. Critical analysis. The study is well structured with clear objectives, controlled experimental design, and strong statistical support. However, limitations include the use of caged bees, which do not fully represent natural hive conditions, and artificial diets instead of real pollen. Additionally, the study assumes microbiota involvement without directly measuring it, which weakens the causal explanation. So for the discussion, the findings highlight the importance of gut microbiota in nutrient digestion and suggest that antibiotic use in beekeeping may negatively affect bee health, especially under poor nutritional conditions. So the study contributes to broader discussions on sustainable beekeeping practices and the need to minimize unnecessary antibiotic use. Conclusion. Overall, the study provides valuable evidence that antibiotics can impair protein digestion in honeybees. Despite some methodological limitations, it offers important insights into the relationship between nutrition, microbiota, and antibiotic use, and is useful for improving beekeeping management practices. So for the Article 4, on the morphology of the digestive system of two Monomorium ant species. Introduction. It is studied by Solis et al. And focuses on the digestive system of Monomorium ants. The species studies is the Monomorium floricola and Monomorium pharaonis, and use light microscopy and scanning electron microscopy. So it examines both larval and adult stages and aims to fill the lack of studies on digestive morphologies in this genus, and important because these ants are common urban pests. So summary, it provides detailed descriptions of digestive organs, compare structure across life stages, larva versus adults, and identifies key anatomical features of foregut, midgut, and hindgut. It also highlights the adaptations and that is related to feeding and digestion, and emphasizes the role of morphology in understanding insect biology, supporting pest management strategies. So for the critical analysis, sa iyang strengths, very detailed and accurate anatomical descriptions, and it uses two of microscopy techniques improves reliability. It also helps fill a research gap in entomology, a organized structure with clear flow of ideas, and useful for both academic and applied fields for pest control. So for the weaknesses, wala siya experimental or functional validation. Conclusions on function are mostly speculative and limited discussion on sample size and geometric variation, and some parts are too technical or difficult to read. So for the discussion, the study is important for understanding insect digestion and provides a baseline for future research and can also support development of better pest control strategies. Also, it highlights a need for more comparative studies across species and experimental studies linking structure to function. So, also, it suggests future research should include behavior and feeding experiments and environmental influences on morphology. Conclusion. The article is informative and scientifically valuable, and it's strong in morphological description and methodology, limited by narrow scope and lack of functional data, and contributes to entomology and pest management knowledge. And also, it serves as a foundation for future studies. The last article, which is the multiple function of Malpighian tubules in insect. The article explains that Malpighian tubules are not only used for waste removal. It highlights their important roles in helping insects survive, such as balancing water, removing harmful substances, and protecting the body. So, for the summary, the article discusses that Malpighian tubules were once known mainly for excretion, similar to human kidneys. However, studies now show that they also help in detoxification and immune response. So these functions allow insects to survive in different environments, especially in dry conditions. So for the critical analysis, the article clearly explains the topic and is well organized, making it easy to understand. It uses different studies to support its claims, which adds reliability. However, since it is based on past studies, it does not include new experiments, and also it also lacks deeper comparison among different insect species. So for the discussion, the article discusses that Malpighian tubules have multiple important roles in insects, not just in waste removal. They also help in regulating water and salt balance, remove toxins, and support the immune system, which are necessary for insect survival in different environments. The information is supported by various studies, making the discussion reliable. But however, the article is limited because it is only a review and does not include new experiments, and it lacks detailed comparison among different insect species.